Now, to understand the difference between mitosis and meiosis, perhaps it is also useful to visualize it again using cookies. Now, okay, you own a cake and a cafe shop. Now, so this is the cookies. Okay, this is the cookies that you have. Okay, now this two cats. Okay, one with the white icing, the other one with the black icings, and they are known as the homologous chromosome. Okay, you can call them homologous cookies. Okay, if you want. Why? Because they have the same length, and then they carry the same. You see, the same patterns or same information. Now, after so what do you want to do now? Now you have a visit from two friends. Yep, and they say, I want the exact copies. Okay, the exact copies you have, as the one on your hand. So this is the one that you have, and you want to produce the exact same copy for them. Now, so what are you going to do now? So to fulfill their wish, you need to use your homologous chromosome here, okay, to act as the template or the mold. Okay, so from these two, then we make a copy of the one with the white icing. At the same time, we also make a copy, the exact copy of the black, okay, of the cookies with the black icing. Now, as you can see here, these two, they are joined together at the central, and they are the exact copy of each other. So these two, we call them chromatids. Okay, now forget about the mirror image here. I know that they are not exactly the same, but just treat, okay, forget about the mirror image. They are just the exact copy of each other. All right, so these two, we call them sister chromatids. And then what about these two? Now, since they are the exact copy of each other, so I call them sister chromatids as well. Now, sister chromatids are joined at the centromere, but the homologous chromosomes do not. Now, so in order to distribute them equally to your friends, now, so what should you do? Now, you ask them, okay, to take a position, and then you place your cookies at the metaphase plate, at the center, so with equal distance, okay, from each other. Then it's time to grab their cookies. Now, so eventually, this friend grabs these two, and the other friend actually grabs the other two, forming two sets of cookies. Now, so this friend, okay, as she wishes just now, she got the exact copy as yours. This friend also has a wish fulfilled. And remember, you own a shop, and on another day, actually, 4 friends of yours come to visit you, and now they are very interested to take your cookies. However, they say, okay, they are too full because they were just after an event. Now they say, we just want either one of that cookies. Okay, we don't want that pair. We only take either one. And then out of these 4, 2 of them say they don't mind. Okay, they will be happier perhaps. Okay, if they are able to have a small taste of different icing. Now, for example, if they get the cookies with white icing, they would like to taste a bit on the cookies with black icing. Okay, and the other one said also the same thing. If she get the black cookies, she would be happier if she would be able to get a taste of the cookies with white icing. Now, so how do you fulfill their demand? Now, so this is their demand. Okay, 4, they say they only wish to get either one of that pair of cookies, and then the other 2 say they wish to have a taste of different types of cookies. Now, so how do you fulfill their demand? Now, so let's come back to this. This is what you have. So we are going to start with what you have. First of all, we use our homologous chromosome to be our mold, to be our template. Then from here, we make an exact copy of the cookies with white icing, and at the same time, the black cookies with the black icing. Now, so we have made exact copy of them. So these two are known as sister chromatids. They are joined together at the centromere. These two, they are also known as sister chromatids, and they are joined at the centromere. Now, how to fulfill the the request, okay, from the other two friends just now, that they wish to have a taste of other types of icing. How do you do that now? So it is right now. Okay, the right moment to switch, okay, the color of the tail. Now, so what happens here was crossing over, exchange of genetic segment between the non-sister chromatids. So these two are non-sister chromatids. All right, now so it is a time to separate them to your four friends I mentioned just now. Now, so this is what you have. So imagine that if you have a cake, okay, a cake which is in a bar shape. Now, how do you cut them into 4 equal portions? Now, so normally this is what we do. Number 1, we are going to cut it at the center to make two equal pieces of cake. Then only, now starting from here, we are going to make another cut here. Yeah, so to separate them into 4 equal portions. Now, so likewise, let's apply it to our cookies right now. So in order to separate them to 4 equally, to 4 of your friends as well, we need to separate the cookies equally. How? We make the first cut at the center. So during anaphase 1, the homologous chromosomes separate to opposite poles. So at this side, you only have the cookies with white icing. On the other side, you only have the cookies with black icing. All right, so they actually inherit. Each cell here actually inherits n number of genetic material. Okay, then we are going to make another cut during meiosis 2. How? We are going to cut it here. So it happens during anaphase 2. Now, so eventually, after telophase 2 and cytokinesis, this is what you have. Now, their requests have been fulfilled. Now, each and every one of them only get either one of their cookies, and then out of these 4, 2 say okay, 2 can have a taste of icing of different colors. All right, so I hope you will have a better understanding about meiosis. Okay, why do you want to cut it at the first? Okay, at the center first. It is because we want to get an equal portion of the cookies or the cakes. Okay, so now if you still have any problem, welcome to see me.